Baik,
Sudah
Pak.
ada berapa Sandra yang jadi pembicaranya? Halo
Saya
Sandra.
masih live streaming
Oke.
itunya, Zoom-nya. Pembicaranya sudah ada 2, Muhammad Fahmi dan
Pak Rezi ya, sudah 2 ya?
iya.
Oke,
Betul.
di-invite aja.
Admit dulu ya. Ini saya pindahkan hostnya ke Bu Yolanda ya, Ibu.
Ya.
Ya, baik. Terima
Nanti kita
kasih.
dikasih co-host, Bu. Bu Sandra sama saya kasih co-host saja.
Oh iya, ya baik.
Sudah terpindah ke Bu Yolanda ya.
Oke.
Mbak Sandra sepertinya ini salah yang satu.
Oh,
Muhammad Fahmi.
sebentar. Untuk bisa memindahkan dari Bu Yolanda.
Oh, oke.
Di waiting room-nya, Bu.
Iya. Ya.
Halo, good morning Pak Reza, good morning Pak Mr. Muhammad Fahmi.
Pagi Bapak Ibu.
Selamat pagi Pak Rezi, selamat pagi Pak Fahmi.
Pagi.
Oh, ada orang Indonesia ya?
Iya.
Sehat Pak Adi, Bu Yolanda.
Iya, selamat
Sehat,
pagi
Pak.
Pak.
Gimana di sana, Pak?
Aman di sini.
Aman.
Iya. Oh Pak Rezi berarti nanti sesi pertama dimulai Pak Rezi, kemudian langsung Q&A ya Pak ya, sesuai yang sudah sama.
Soalnya nyambi di lab juga, itu nggak bisa saya tinggal.
Oh gitu, iya Pak. Nah, nanti insya Allah bisa cepat.
Ini Pak Reza yang pertama mau coba share screen dulu, Pak.
Oh boleh.
Ya. Lalu setelah itu Pak Muhammad Fahmi ya. Lagi Pak Muhammad Fahmi.
Iya, ini untuk Pak Fahmi ini belum connect ini Pak, apa? Mic-nya.
Oh, belum connect?
Iya, mic-nya.
Oke.
Aman Pak ya?
Oke Pak, keren Pak.
Aman, sudah Pak.
Sudah terlihat jelas dan keren. Oke, jadi ini nanti moderatornya Bu Yolanda, Pak Rezi dan Pak Muhammad Fahmi. Nanti saya akan buka, lalu akan saya oper ke Bu Yolanda. Masing-masing pembicara 20 menit ya Bu Yolanda. Kalau lebih paling lama 40 menit Pak.
40 menit ya, siap Pak.
Siap.
Nah, itu karena Pak Rezi ada agenda lain, maka langsung Q&A. Gitu.
Iya.
Nanti setelah Q&A Pak Rezi, lanjut ke Pak Muhammad Fahmi.
Iya. Ya, baik Pak.
Jadi nanti Pak Johan pembicaranya yang dari Korea dan Jepang satunya ya Pak Bu ya.
Iya betul Pak. Betul. Untuk yang ini juga sama dari Korea dan Jepang Pak, namun diaspora ya, diaspora yang besar. Ya.
Tapi kalau ini dua-dua dari Indonesia, kalau yang satu kayaknya satu India, satu
dari Afrika Pak ya.
Afrika kayaknya.
Oke, kita tunggu sebentar ya Pak Rezi, kita akan mulai nanti.
Kita mulai jam 8 aja.
Iya.
Nanti ini kita another 10 minutes. We will be back another 10 minutes. Kalau mau ngopi-ngopi dulu nggak apa-apa. Oke Pak, see you in 10 minutes Pak.
Oke.
Bu itu nanti kurang 5 menit atau kurang kurang 5 menit semua peserta bisa aja Bu.
Iya, baik Pak. Nanti langsung saya eksepsi.
Oke. Dan dihilangkan untuk waiting room-nya aja Bu. Kalau sudah.
Iya.
Oke Bu.
Ya, terima kasih Pak. Iya. Selamat pagi Pak Fahmi. Sudah connect kah ininya mic-nya? Halo? Tes.
Halo.
Halo, sudah muncul ya Pak?
Halo, ya sudah. Halo, pagi.
Selamat pagi. Thank you Pak Fahmi. Jadi nanti sesuai dengan rundown nanti akan dimulai Pak Rezi dulu.
Yeah.
Kemudian karena Pak Rezi nanti setelah ini langsung ada agenda lain, jadi setelah itu akan ada Q&A, kemudian langsung lanjut ke Pak Fahmi seperti itu ya Pak ya.
Oke, iya, terima kasih. Oke,
Itu langsung kembali dilanjut dengan QA.
ya, baik.
Oke, kita akan mulai mungkin 9 menit lagi ya Bapak-Bapak. Jadi setelah ini akan saya invite semua seluruh attendees seperti itu, kemudian kita akan opening sebentar, kemudian kita lanjut. Oke, terima kasih. Silakan jika mau ambil kopi dulu.
Good morning, everyone. We will start in 5 minutes. 8 o'clock sharp. Okay. See you in 5 minutes, guys.
Okay, thank you.
Hello, good afternoon, good morning, everyone. How are you?
Good morning.
Can you hear my voice clearly, guys?
Yeah, it's clear.
Okay. So my name is Aditya Jakusuma. I'm the chairperson of the Ubaya Global Webinar 2023. I will be the MC for this webinar for the next 2 hours for the Faculty of Biotechnology. This morning, we are very glad that we are now come to the last session of our global webinar, and it is hosted by Faculty of Biotechnology. And today we will listen about artificial intelligence in biotechnology research and integrated approach. We are very honored today that we have 4 speakers from 2 countries. One is Japan and another one is South Korea. The first speaker from Korea is Pak Resi Eko, Mr. Resi Eko Caraka, PhD. Good morning, Pak.
Morning, sir.
He is a senior research fellow from Ulsan National Institute of Science and Technology, Korea. He is also visiting professor at Bukyong National University, Korea, and also brain researcher. Today, he will deliver a topic of using spatial big data and cognitive analysis to prognosticate malaria prevalence across Papua and West Papua, Indonesia. And then the second speaker who will talk today is Mr. Muhammad Fahmi, DSC. Good morning, Mr. Muhammad Fahmi.
Morning. Hello.
Hello. He is from Research Institute for Humanity and Nature, Kyoto, Japan. So you'll have Korea first and then Japan. And Mr. Muhammad Fahmi will talk about boosting AI and biotechnology research with Nextflow, a comprehensive workflow management solution. And later we will also have Dr. Satya Murthy from Insilicogen in South Korea, and then from University of Tsukuba, Japan. The speaker would be Oscar Vitere Italo, PhD. And to moderate this event, we have Ibu or Mr. or Dr. Yolanda Antonius. Good morning.
Hello, good morning, Mr. Adi.
Ready for the event, Bu?
Yeah, we are ready.
Okay.
Thank you.
Yeah. So the speakers would speak around 20 until 40 minutes, and then it will be continued with Q&A session. And during the Q&A, you can directly ask or speak, or you can type on the chat section. It's up to you. Which one is convenient for you, just do it. Okay. So without further ado, now I give the time for Dr. Yolanda Antonius to moderate this session. Please give applause from the Zoom reaction.
Yeah, thank you, Mr. Adi. Hello, everyone. My name is Yolanda, and I'm responsible as a moderator for guiding this session. First of all, I would like to welcome all the speakers and attendees in this webinar. And just for information that this webinar is attended by the students and also live streamed through the YouTube online. And again, I would like to acknowledge for all the speakers for taking your valuable time to be here and share about the update knowledge related to biotechnology. Before we are going to the presentation, I just want to inform you that this webinar will be done in 2 big sessions, as Mr. Adi mentioned before. And for each session will be presented by 2 speakers, which followed by Q&A session. So now for the first speaker, we are welcoming Mr. Rezi Eko Caraka. Hello, Mr. Rezi. Good morning.
안녕하세요. Good morning.
Yeah, 안녕하세요.
안녕하세요, 여러분.
Yeah. Okay. Yeah. Recently, Mr. Rezi, he is a researcher in Research Center for Data Information Science Stream, and he is also a adjunct lecturer in several universities in Indonesia. Moreover, he had a lot of experiences as a postdoctoral fellow in Korea, and also he is a top 2% scientist in the category for artificial intelligence. So it's an honor for us to invite you here. So we really hope that we will have valuable knowledge and inspiration from your presentation. So now please welcome Mr. Rezi. Yeah, the time is yours. Please.
Thank you, Dr. Yolanda. Let me start share screen. Okay.
Okay, it's good.
Okay. Very good morning, audience, faculty member Universitas Surabaya, and also honor to Dr. Yolanda and also Dr. Muhammad Fahmi. And thank you for guys to coming for this practice agenda, isn't it? So today's presentations, I will share part of my research in South Korea. We try to using kind of the big data and also the cognitive analysis to prognosticate malaria across Papua and also West Papua, Indonesia. And it's our agenda today. I try to explain to you, maybe most of you are not familiar with remote sensing. So I try how we make analysis to remote sensing and how we can harvest data from the satellite to topic about the health. Okay, my name is Rezi, so you can just call me Mas Rezi. So I hold some of the non-tenured faculty member like University of Indonesia, and also Universitas Pajajaran, and also ITS. And then currently I serve as researchers in Indonesia. I just joined by last year, and also hold some of research fellow in South Korea like the UNIST in Ulsan and also Pukyong National University. So here my research is mostly about SDGs. So maybe most of you knows about SDGs. SDGs is some of our ambitious topics that we want to reach start from 2015 until 2030. So it consists of the 70th topic, start from no poverty until the partnership for the goals. Of course, if you're talking about the big data, the data can be everywhere, like the cone data and also the image data set. So I suggest you to learn the basic data science, and then you can join with us to maximize the benefit to SDGs. Here, to start this discussion, guys, maybe you can start what are your passion. So maybe you will more familiar about how we can tackle the no poverty, and also how if we can tackle the zero hunger. So actually the visions of SDGs here, we want to maximize the effectiveness of all forms of the cooperations for development, for the shared benefits of people, planet, prosperity, and peace. So some of researchers says that it would be as the pentahelix collaboration within we as the academicians, professional researchers, of course, and also here we have the private sector, of course, to benefit it, and also we have the NGO people and also media itself. So it could be as the pentahelix. Here, Indonesia is also trying to reach the SDGs by some of the policies, of course, start from 2015, the predictions make the increasing of awareness to our people. So we working together to make our people aware about the SDGs, start from 2015. And also 2016 presidential decree on SDGs draft, we make the blueprint. And then 2017 officially signed the presidential decree. And start from 2018 until 2030, of course, we will working together how we can maximize our potential to make the SDGs really good. Of course, if we compare about Indonesia and Malaysia, we still have much homework from this figure. Here, we see that Malaysia is more steps ahead if you're talking about the how we battling about the no poverty. So here, you can see in Indonesia about the increasing, about the how we stagnant in point 14, about the life below water. So very sadly, because most of the areas in Indonesia could be covered by water. So we we can working together how we can maximize the point 14, of course. So it's we can compare that Malaysia is more step ahead than Indonesia. And talking to the big data, talking to the data science, of course, you can do anything. We can harvest the data from the social media, like the Twitter, like the Facebook, and also if you want to analyze the economics issues, of course, you can harvest your data from data stream. You can harvest your data from many sources toward the cryptocurrency. And here, toward the multimedia data, we have the pictures data to make the segmentation, to make the classification, so what on that. And then here, as the researcher, of course, maybe some of you have experience to measuring the survey and also questionnaire. And then we can harvest our data from the satellite. Yeah, these are the data sources from now. And start, are we talking about the remote sensing? What is actually about remote sensing? So I'm typical of researchers is most believe what I see than what I feel. So remote sensing is part of the data science area that we can harvest our data from the satellite. So the data set that we can analyze is of course from the NASA. So NASA publish some of the data set that we can do the analysis toward the SDGs. So here the definition is the NASA, and also we also have the satellite, of course, to observe our Earth with the planetary bodies via remote sensor on satellite. Yeah, these are some of the advantages of the remote sensing. Here, if you go to the NASA website, of course, they have some of the category towards the data set, like the start from the first atmosphere. Atmosphere is data set from the geophysical forces, including the clouds, aerosol, precipitations, and also others meteorology and climatology data set. And of course, we have the biosphere, cryosphere, until the terrestrial hydrosphere. So here, that if you want to make the analysis that I'm most familiar with the human dimension, wait, my alarm is turned on. Hold on for 2 minutes.
Yes, please.
Sorry, wait.
So for the attendees, if you have a question for the presentation of Mr. Lazy, so please don't hesitate to write the question to the chat room, or maybe you can chat in private for me. Thank you. Please.
So I continue because
Yeah.
I should turn off some of the instrument. That's why I turn off the alarm. So we moving to the dataset from human dimension. So I'm most popular with this area. So human dimensions means that we collectively harvest dataset from NASA, including the government agencies like the census, data disease, species diversity, and also some of similar data. So from the Indonesia side, of course, we have the official statistic or BPS. Yeah, we can combine how if we do the analysis from the BPS dataset, and also we combine within the satellite dataset. So we can maximize all the benefits in the SDGs. Here, that we try to reach down the Papua problem, start from the health issues, start from the hazard issues. Of course, if you're talking about the hazard, it can be divided by 3 aspects, guys, like the natural hazard, like the volcanoes, tsunamis, flood, and also we have the non-natural hazard, like the disease, like the COVID-19, tuberculosis, malaria, of course. And then also we have the social hazard, like the protest, demo. Yeah, if you're talking the hazard, it can be from the many aspects. Here, I went to Papua from the 2014 from the to the social experience. Yeah, Papua as the one of the regions that many have the potential, like the species diversity, and also how the atmosphere there, that we can have the many good species and also biology. Of course, the sadness that in Papua they have epidemic disease like the leprosy and also malaria and also Yos disease. So if talking about the health issues in the Papua, of course, we still have the much homework that we can solving these issues. So we try to make the spatial mapping to the malaria cases in Papua and also West Papua. So from data set here that we use the remote sensing, of course, by consisting of the mean solar radar surface, and also we collect data set from the water vapor, total rainfall, net radiations, vegetation index, and also land surface temperature. And then, as I mentioned before, that from the research that we try to combine the satellite data set to the Susanas data set from the BPS, the Republic of Indonesia, like the disposal phases, like the drinking water source, like the how the Papuans can have the lifestyle of the hand washing facility, and also waste containment, and also the malaria cases around Papua and also West Papua. And then here the maps of the Papua from the regions within the Papua and also West Papua. Yeah, after that we do analysis that of course the highest cases, the malaria in Papua should be in Mimika. So we can get the analysis how to battling the malaria around the West Papua and also the Papua itself. And then start from data analysis. For the basic of data analysis can be divided by three aspects, like the regression, post, clustering, and also classification, the basic things. So if you want to more detail about the many methods or approaches in cluster, you can read the detail from my paper here, that I try to make the some of the procedure that if you want to handling your data sets using the hierarchical cluster, and also how if you want to using the clustering about the non-hierarchical, and also how if data set is hard dimension. Of course, you can using the machine learning, also fast computing techniques. That here we develop our own methods called be NASPA class. Yeah, NASPA class is our developed methods that have already published on our software, also our library. If you want to using these methods, of course, you can do. We also provide our manuals and also the practical of the hands-on. Of course, you can try by yourself. How if you want to analyze using our methods NASPA class. Here, you can go through to the our software, also our library. Here, we provide some of the description, so the manual, so you can analyze this by using your own data set, of course. So we bring many novelties here. Like first of all, the basic of NASPA class is for the form graphically using the fuzzy. So after we study more detail about the fuzzy itself, we realized that the fuzzy have some disadvantage. Like if we do the mathematical things, yeah, it can be concerned from the Gaussian discriminant analysis. So these are disadvantages of the fuzzy GVC. And then we try to cover about the methods that we can using kind of the optimization. Here, from our optimizations, we propose 7 optimizations, like the we mimicking from the natural experience, like the artificial bee colony, from the particle swarm optimizations, Harris-Hawk optimization, random walk. Yeah. So if you want to using our own methods, of course, we provide the many single combinations like the NASPA class using the ABC, bee colony, or you can using our NASPA class using the flower pollination algorithm. Yeah. Here we have the 7 combinations of optimization. And also, if you're familiar with regression, you can using the R-square to compare the accuracy, but we can do that on the clustering. Some of our researchers are using kind of combination like the silhouette index, then index. So, but here we also using some of the 7 combinations like the PC or the particle coefficient. We have the CE until the quant index. So we have provide 7 algorithm optimization, and also we provide 7 criteria to evaluations this method. So this method can be beneficial to analysis, because if you're using kind of the machine learning, deep learning, of course, it could be as the black box model, you can do as the interpretation. But during last 2015 or 2016, if you read more detail about the statistical journal, most of researchers move to analysis by using the glass box model. So glass box model can be using how we can get the higher accuracy, and also it can be benefit to the reasoning for your problem. Here, the basic of the ABC method using NASPA class, that of course, because this data could be spatial and geographically. And here D, we have the distance within the location A and also location B. Also, PG and XI should be X the our independent variable. So the different things that using regression, of course, you have two types of the variable: dependent variable as Y, and also independent variable as X. So you have keep in mind, in the clustering, we only using every single X variable, X1, X2, X3 until Xn. So it's different within the regressions and also clustering. In clustering, we don't need from the dependent variable. We just need from the independent variable or every single X variable. So if you understand about the how the B can be survived to our nature, of course, B can be divided by three groups, guys, like the worker bees, search bees, and also scout bees. So try some thinking that some of these maybe we will fly through the flower. Yeah, here that's why here I try to denote that as the phi. So phi as the foot position. So most of bee will be fly together. They in grouping to go through the flower. So we can analyze this how every single bee can be as our base parameter. So we can try to capture which bees can be make our models will be high accuracy. So start from the analysis, from the calculating the malaria. So we using the basic equations, like the cases divided by populations, and also multiply by thousands area. So from the data set that I used before, because the goals of the data science that we want to maximize our benefit, that's why we try to using many variables here, like the remote sensing, and also from the BPS side. So here, if you want to select which variable can be benefit to analysis, so I suggest you can using the MSA or measuring sampling adequacy, and then you can choose your own threshold. So here I'm just using my own threshold like the 0.6. So once the variable cannot be reached but the 0.6, of course we should delete that information. So it depends on you. So most of the researchers are using the 0.3, most of the researchers also using 0.4 and also 0.5. So it's kind of the similar like the significance level. So you can using by your own judgment. So the reasons they are using the 0.6, of course, I try to confidence that the probability from the similarity in Papua and West Papua around 60 similarities. Yeah. So from this criteria, that the most significant variables should be from the total precipitations from the breeding of malaria. Also, we have the total evaporations, and also, of course, about the washing water towards around Papua and also West Papua, and also the drinking water is so similar around the Papua, and also about the hand washing facilities, fishing disposal, and also defection facilities around the Papua. So after we run lots of the simulation, start from the coefficients 2 until the 5 from the clustering, and also combinations of the formations every single bees, like the, for example, how we using 100 bees. Of course, we have this criteria. So from these research that we try to make the clustering by using 3 clusters around Papua. Here, you can use, you can justify with a different set of a color here, like the blue, like red, and also the green within the cluster. So the research that we get, that we have already successful to make the spatial towards in Papua, and then this paper have already accepted in scientific report. Maybe we will publish by this week. So maybe if you want to read more detail, of course, I can send this paper to you. And of course, if you want to the data set, of course, just ask me, just let me know. And then we save our future research that we interest to see how to capture every single species from the malaria in Papua. like the albopictus, and also Aegypti, Anopheles, and another species from the malaria side. So these are we save for the future that we want to working more detail again to capture not from the anthropology side, but also from the environment itself. So maybe that's all for my presentations. Of course, if you have more also another questions, just let me know. You can reach my email toward rezi.eco.charaka@brain.go.id. Of course, if you want to the single of the coding or syntax, just let me know. I'm happy to share of my coding. Okay, thank you very much, Dr. Yolanda.
Yeah, thank you, Mr. Rezi, for the presentation. From this, we know that data spatial, for remote sensing is really essential for us to create the mapping for the disease or any other parameter that we want to see. Okay, so we are moving to the Q&A session. So please, anyone, if you have any question, so you can raise your hand or you can drop the question into the chat room. So first, I see here Mr. Maulana Malik Nusilo. So please, you can unmute yourself and then you can ask to Mr. Rezi, please.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Thank you for the presentation, Mr. Rezi. Long time no see, Mr. Rezi. So, I want to ask about because you show a lot of data, right? And then how if public want to access such data? Because I don't know, in Indonesia we have some kind of initiative to share such kind of data for us. So maybe we can reanalyze, or maybe we can do some research based on this data. And I don't know, did BRIN nowadays have the such repository for this kind of the biological data sets for us, researchers in Indonesia will use for it. Thank you.
Okay, hi, Sir Maulana Nungtanusi. Actually, I'm not familiar with the BRIN site. Actually, I just joined by last 1 year. And then for my site, that mostly I also publish my dataset, Mr. Maulana. So from the end of the part of the paper, of course, I also mentioned my GitHub. So I'm more than open to share my dataset and also the coding. But the question is that if you're looking for the sum of dataset, of course you can reach by different type of the ministries, isn't it? Because as we know that we also have the one data. So I'm also not sure that the data is the update yet. So yeah, that's my answer, Maulana.
Okay, thank you for your information. So, but the BRIN currently don't have the repository yet for this.
It should be have, but maybe I don't know the detail from that, Mr. Maulana.
Okay.
I just new join from the BRIN side.
Okay, thank you very much then.
Yeah, thank you, Mr. Maulana, and thank you, Mr. Rezi. So we are now going to the second question. It's from the student, from Eleonora. So maybe I will just read the question, Mr. Rezi. Okay, so the question is, she asks about there any information about volcanology, topography, rainfall, and other geographic that you present in your presentation. And the question is, do those information affect to the malaria spreadness in Papua? Maybe she just curious about the effect from the information from non-biotic information to the malaria spreadness.
Yeah, thank you for the questions, Dr. Yolanda. Of course, that here we just actually try to using many variables. That so here that you can see that from the side of the remote sensing from the satellite and also side of the BPS, so we can see which variable is most significant. Of course, from our research that we have proof that here that the variable that we can using from the breeding from the Maria set, like this mean temperature and also evaporations. Also from this research that we also recognize some of the disadvantage because we realized that there are many species from the Maria. So that's why we try to recalculate. We also try to redefining from this research data Yolanda. That's why we left the future research that we interested to looking more detail every single species. Maybe it will be different, Dr. Yolanda. Yeah.
Yeah, okay, thank you for the answer. So maybe I will just more curious about the explanation that you state that you want to dig more deeply about information so you can get the data that more detailed. So is there any specific criteria for the data so we can use it for the detailed mapping of the malaria spreadness?
Pardon?
Is there any specific criteria for the data?
Yeah, of course. We're looking for that, Dr. Yolanda. That's why from this, start from the last month, we working from the epidemiology side. That we are moving from the working from the another expertise, Dr. Yolanda. That's why from this beginning research that we not put the epidemiology to our research.
Okay, but you are planning to put it into the next data.
Yeah, that's why this vendor of data science, of course, we not very familiar with the expert system. Yeah, we only recognize which variable is will be a benefit. Yeah. So from this research that we have already make the clear, so we should using the ABC variable, and then we will working together from the public health side, the epidemiologists. Yeah.
Yeah. Okay. Thank you, Mr. Rezi. So maybe I also have one more question about the restriction about the data because I'm still curious about this. Yeah. So is there any restriction data that maybe that's the data that we cannot use, or how about the consideration? What kind of data that you want to or you plan to use to enrich your analysis?
Yeah, from data set also, I try to with my friend. We are from he working on the BPS side, so we try to see what that variable that we can use to that data Yolanda. So we also try to study more deeply.
Yeah.
From that data set.
Yeah, that's interesting, and we still have so many questions that need to answer, yeah, Mr. Rezi.
Yeah,
so I hope that your research will be successful. Okay, thank you, Mr. Rezi. So for anyone, please, if you still have any question, so you can raise your hand or maybe you can drop the question in the chat box, please. Okay. If there is no question anymore, or maybe later you just remember about the question, so please just put your question. You can send it to me, or maybe you can send email to Mr. Rezi. Is that okay, Mr. Rezi?
Oh, sure.
Yeah, okay, because for remote sensing in our university, it's related new, so we are just curious. So thank you for Mr. Rezi for spending your time here to share and to update the knowledge blocks. Thank you, Mr. Rezi.
Thank you.
Thank you. Okay, everyone. So now we are going to the second speaker. And here we are welcoming Mr. Muhammad Fahmi, DSC. Hello, Mr. Fahmi.
Hello. Can you hear me?
Yeah, it's good.
Okay.
Yeah. Okay, maybe I will give the short, a brief about the introduction. So Mr. Fahmi, he is a researcher in Research Institute for Humanity and Nature in Kyoto, Japan. And he is also a visiting researcher at the Ritsumeikan University in Japan. And like we know, he had many research publications related to the bioinformatics. So now please welcome Mr. Muhammad Fahmi. So
thank you so much for your kind introduction, Miss Yola. So long time no see, I think. Oh, we see. We meet last month. Yeah. Okay. Yes. Today, my talk is focusing on Nextflow, workflow manager. That I think nowadays has become essential to master. I will give an introduction and basic knowledge on what next Luis. Additionally, I will talk about my experience in using this workflow manager for my research project in RIN. And I think mastering this workflow manager can boost the research in AI or AI-powered biotechnology research. And talking about the connection between biotechnology and computer science, I think we are talking about bioinformatics. As you know, bioinformatics is an emerging field right now that I think most of the scientific disciplines in biology, the analysis rely on the computational power of bioinformatics. As you know, there are many disciplines, for example, molecular biology, genetics, omics, and pharmaceutical science and AI that I think has got some advantage from the bioinformatics fields. And if you're familiar with bioinformatics, you know that you often in situation where you are trying to write complex pipelines or analysis. So this is the example of a pipelines in doing genome annotation of Mycobacterium tuberculosis NGS data. So from the NGS data, usually you'll get FASTQ data, for example, from Illumina. And from these pipelines, so the author here performing quality control using FastQC. And after that, they are cutting the bad quality of sequence using chemometrics. And after that, they're using the downstream analysis. For example, here they use Kraken to perform the combination checks and SPIDS to perform the de novo assembly. And for Fourier calling and filtering, they use SUM tools and PCF tools and etc. As you see here, there are so many tools that they use to annotate the genome of the Mycobacterium tuberculosis data. So I think it is more than 10 of tools that they use here. And if you run the analysis traditionally, or even worse, like one by one using UI tools, I think you will take so much time to perform the analysis. So in here, I think you need to write the pipeline. So for the traditional pipeline, I think if you are familiar with Linux, so I think you will use Bash shell to write the pipelines. Or if you are familiar with Python, so you can write it in Python to write to embed the tools in all of these tools in the Python, so you can run the analysis automatically. So this is, I think it's not so complex compared to another pipeline. So this is the example of very complex pipelines in bioinformatics. So here, Stanbis developed software called Companions to annotate the parasite genome. So in these pipelines here is like very complex. They use like 70 process and like the combination of 55 external scripts and like 39 software tools. So if you run it manually, I think you need to install one by one of the software tools in your local PC. So it has like 39 plus 55 scripts. So I think it's like around 90 tools or software that you will use. And it will take a lot of time. You will perform this pipeline manually, or it takes forever because you will get lost in the middle of this process if you run it manually. So that's why I think we need managers to solve our problem in dealing with the bioinformatics pipelines. So this is the comparison of the traditional pipelines and workflow manager. So in traditional pipeline, if you are familiar with bash shells or scripts such as Python and etc., this is the example of performing transcript expression quantification using FASTQ and reference sequence as the input. And here there are three kind of tools that they use: FastQC, Salmons for quality control, index creation, and quantification. And in the transitional pipeline, so I think you will need to install the requirements. For example, FastQC and Salmon, you need to install it first in your local PC, and then you will need to write the pipelines code, and then you will execute in the local PC, for example, in this traditional pipelines. But in the workflow manager, you can embed your requirements of FastQC and Salmon in your pipelines code, so you don't need to install it one by one, and you can use containerization such as Docker if you are familiar, and then you can run or scale up your execution not only on your local PC. You can also deploy your pipelines in the high performing computer and cloud, for example in AWS and etc. Easily. So here what I want to talk this is Nextflow. Nextflow is an open source workflow management systems that allows you to create, run, and share a computational pipeline. So this is a DSL program or domain specific language. They are built on top of Java and Groovy, but within this programming language, you can embed your any other scripts. For example, if you are familiar with R or Python or Bash, you can embed your writing code into this workflow manager. And they also can be connected to Conda, Docker, and Singularity, and any other containerization. So you don't need to install all requirements in your local PC. So you just need to define what is the tools that you want to use in Docker, for example, and then they will just run it easily. And they have version control. They can be controlled using GitHub, and then they can be deployed in every major platform. For example, in AWS, Slurms, Azure, and etc. You can also run this kind of pipelines in your local PC. So this is the step-by-step to install the Nextflow. So I think you need to use a POSIX compatible system, for example, Linux or OS X. But if you are familiar with the Windows, I think in Windows right now you can install WSL, Windows Subsystem Linux. So you can also run Linux in your Windows right now. I think it's very easily, and I think you can also perform this to use this Nextflow in your Windows system. So you just need Java. So make sure to have the Java version in your system, and then just perform this command line, and then you can launch the Nextflow by running the program. Is like Nextflow run, and then your Nextflow scripts. And this is the example in the bioinformatics. This is the tax example in the command line. So in BWA-MIM, if you have a NGS sequence and you want to align it to a reference sequence. So for example, here, your sequence called sample FASTQ, and then you have a reference called reference FASTA. To perform the alignment, you can use this BWA-MIM, and then they are piping these some tools to sort the aligned NGS data. So this works in your Linux or OS X, for example. But so if the comment is works, so you don't need to like change this comment. So in Nextflow, you can just embed this command line into the Nextflow. So this is the example when you want to embed your comments into the Nextflow. This is the process called align samples. So this is the script that we saw previously, BWMM. So in this script, you can embed your tags of bioinformatics tools, and you in Nextflow, there are some kind of system called input and output. So this is the input is your sample.fastq and your reference .fasta, and the output is called sample BAM in here. So after you define the input and output, you can connect the output from the align sample, for example, to another process called index sample here using some tools index. So you can connecting one tools to another using channel. For example, here the output is called BEM channel. You can use this as the input in the index sample here. So with this connection, you can like create the pipelines of a complex pipelines. If you are using like more than 2 or 3 or even was like a 90 kind of tools in bioinformatics, you can make the pipelines of that complex pipelines. And in Nextflow, another advantage is you can do parallelization. So in Nextflow, if you have more than one data to analyze, for example, here in the aligning, if you have like 100 or like 1,000 samples, you can perform parallelization very easily in Nextflow. So this is the example of parallelization. So in one process, if you have like 100 or like 20 data to analyze, they can perform the parallelization. So it can speed up your analysis. So this is the example of parallelization in Nextflow. If you have a folder called data, and within that folder you have, for example, sample.fastq. If you run this data into Nextflow, you will only run once because the input is only once. But if you have like 100 or like 1,000 data within the folder data in FASTQ format, by using asterisk here or called wildcards, you can run the quality control in parallels. So if you have like a 100, you maybe can run the like, I think it depends on your computational power. If your computer can run like a 10 or 20 program in parallel, they will be run in like a 20 or 10 in parallel. So it will like speed up your analysis if you have very big data. And you can also adjust like how much parallelization that you want. You can embed a script in here. Like if you want just like a 10 in parallel for this kind of process, you can define it easily in this basic script. So this is just the introduction of the script. So if you want to learn more about Nextflow, you can check in the nextflow.io. I think they have so many documentation that if you like can kind of master this kind of workflow manager, I think you will have some kind of like very powerful tools to have in the bioinformatics fields. And as I said previously, so Nextflow can be run in your local PC, and it also can be run in your if you have some kind of server or SPC or cluster computer, you can deploy your program to run it easily in the cluster computers. It also can be run in the clouds. For example, if you have AWS Batch and S3, you can deploy your program easily in the clouds. For example, here in AWS Batch, if you have so many data to run, you can deploy, and they will submitting the jobs automatically, and they will save the result automatically to your S3 in the AWS. So Nextflow right now, they also have some kind of community that they build so many pipelines in open source. So if you don't need to master your the Nextflow programming to use it. You can just like download the pipelines that the community has built in the GitHub. You just can like download the pipeline and run for a specific kind of analysis. So this is the example of Nextflow pipelines in NF Core for RNA-seq analysis. So this is the pipeline of the RNA-seq analysis. They performing the quality control, alignment, and then etc. And in here you can see they are very complex, but you don't need to write the pipelines one by one. You just need to download these pipelines, and then if you have FASTQ data, you can just like perform Nextflow run RNA-seq, and then your input in FastQ, and then they will perform the pipelines automatically, and we will get many outputs. For example, here GTF, FASTA, TSV, BEM, and the reports here. So you don't need to build the pipeline; you just can like download it. So this is another example of next NF Core Eager. So in here, Eager is a kind of genome annotation for ancient DNA. So yes, again, you can just download these pipelines in the NF Core, and then you can just run the program. So you don't need to build the pipelines, these complex pipelines. So for AI, there is like emerging tools called AlphaFolds. So you can like predict the folding of protein using AlphaFolds. So if you have so many proteins that you want to predict the structure, like 1,000 or like 10,000 of protein structure, you can just put your list of protein that you want to predict in the CSV, and then just input it in this pipelines called protein fold, and then you can just run it automatically. So you just run, and then you can just watch it to performing this kind of analysis. So that is the introduction of Nextflow. I think you can check it the complete picture of Nextflow in their website. I think they have so many documentation and YouTube to learn. And so after this, I will talk about my projects in RIN, and in which I use Nextflow to perform my analysis. So in RIN, I do a research project called Investigation on the Impact of Global Environmental Change to the Development of Future Healthcare. So in here, my focus is my object of research is natural product. So natural products refer to chemical compounds that are produced by living organisms, such as plants, animals, microorganisms. These compounds are often characterized by their unique chemical structure and are used in a wide range of applications, including medicine, agriculture, and industry. And natural products have been used for medical purposes for thousands of years. This is the example of Egyptian medical papyrus of herbal knowledge dating to like 1550 before Christ. So human have used natural products a long time ago. And we have so many natural products that we have identified right now. For example, natural product can be classified into several categories based on their chemical structure, such as alkaloid, terpenoid, flavonoid, and polyketides. And these compounds are often biologically active and can have therapeutic properties such as anti-cancer, anti-inflammatory, or anti-microbial effects. For example, here is paxillatals. Paxillatals is plant-based natural products that is used for cancer. So this is another example of natural products. So this is the venom from Polybia paulista, which venom can be used to destroy cancer cells. So this is the drugs, the approved drugs from 1981 to 2014. So based on this pie chart, biological molecule, unaltered natural product, and botanical drug, and natural product derivative, they are like some kind of around like 45, almost 50, almost half of all approved drugs right now. It came from natural products. So they are very important resource for the development of future healthcare. But they rely on the biodiversity. So natural product is came from biodiversity. So the plants, animals, and etc. But right now our biodiversity is in jeopardized. They are losing right now because of the pathogenic effects. So this is the example in my research projects from Kanemoto-san's. They built some kind of global hotspots of species threat linked to consumption in the United States. So the consumption here have so many effects on the global scales that they even people that live here, they can destroy many biodiversity in another regions. For example, in Indonesia here, the lands and the sea, the here is destroyed by the consumption in United States. So they are importing many products from our resource here. And right now, the biodiversity is losing. This is the index value, Global Living Planet Index, that we are right now have some kind of decreasing in the index value here. And we need to kind of like counter the issue. If we don't counter the issue, our diversity will be lost, and the source of natural project will also lost, and it will threaten our next future healthcare. So we need to identify the problem, and then we should counter the problem here. So this is my research. So in here, I want to measure the global pattern of natural products. So by measuring biodiversity in here, so the aspect of biodiversity can be measured in three aspects. You can measure the species richness using taxonomic diversity, and there's also another kind of method that you can measure in the evolutionary aspects using phylogenetic diversity, and the functional aspect using functional diversity. In here, I use phylogenetic diversity by using the markers that is related to natural products. In here, I use 9 markers of enzyme family that I use to identify the global pattern of natural product here. So from these 9 family markers, I use this to develop a phylogenetic tree, and then I map the diversity distribution in the spatial maps. So this is the method that I've used. So in here, I use genome and transcriptome data. So my object of research is woody plants, because woody plants can be easily mapped in the spatial mapping. So I download all genome or transcriptome data that is available in the NCBI. So for woody plants, they have around 55 species that have a genome, assembled genome. And if they have assembled transcriptome, I also download their assembled transcriptome. It has like 399 species that has assembled transcriptome, and there are so many raw transcriptome, which means transcriptome that has not been assembled in SRA. I retrieve like 990 species transcriptome. For this data, it is so big that I think it's around 100 terabytes. So because the data is so big and I need to perform a complex kind of analysis, so to speed up my analysis, I build a Nextflow pipeline so I can perform a parallelization for my analysis. So in here, I perform a quality control using these 3 software, and then perform de novo assembly using these 3 software, and then evaluate the completeness of my assembly using Basco, and then mapping the distribution using this software, Maxen and GBEF, and then performing sequence similarity search, phylogenetic tree, and then calculate the phylogenetic diversity, and then maps the phylogenetic diversity to the spatial distribution of the species. So this is the data. This is the distribution of data that I retrieved. So for the genome and transcriptome data that is available in the NCBI right now, mostly came from China. I think because they have some kind of like a project right now called 1000 genome profile. So from that project, they produce so many genome or transcriptome data. So yeah, mostly my data came from China, and for Indonesia, I think it is very few. I think it's less than 15 here. So this is the completeness of the my genome and transcriptome data. Comparing the genome and transcriptome data to the reference, I can see the completeness of my genome, whether they are not complete or there are so many missing gene from my assembled transcriptome. So I set like around 55% for the cutoff. So if the quality of the genome is less than this 55%, I remove this data and I only use this data here. So this is the phylogenetic profiling. If you can see here, there's a very small text in here. So in here, in the right side is the species. This is a clustering, and the downside in here is the gene. So the white dot here means the absence of the gene. So for example, in here, there is a white dots here. For this gene in this species is absence, and for the black dots means presence. So this genes in this species is present. So as you can see here, they are clustered into 3 clusters. So for the gene, there are class A, B, and C, and there's a class B that is conserved. So the gene can be always present in all species. So I use these genes for the marker. So it around like 42 genes here. So from this cluster, I check the function of their genes. So using gene ontology. So for the conserved genes, they have functions such as secondary metabolites, phenylpropanate, and etc. And this function is, I think, related to the natural products. So using these genes as the marker, I think, is reliable. And from these genes, I think there are like 42 genes. I built a phylogenomic tree using these 42 genes in the 1,000 species. And this is the phylogenomic tree that I've built. And as you can see here, the reliability of this tree is very reliable because mostly colored in purple. So purple means they have 100 in reliability and the maximum likelihood. So as you can see here, the reliability of this tree is very high. So from this tree, I check the branch lengths and performing the phylogenetic diversity analysis, and then mapping it to the spatial distribution. So this is the global presence of woody plants that has natural products. So there is a left and right. So for the left, I use a reference tree from the KBOTB that they use a traditional taxonomic marker, for example, mitochondrial DNA to build the tree. And this is my tree that is built from the medicinally associated enzymes. And comparing the diversity of these 2 trees, as we can see, there are some region that has potential for the medicinal aspects. So I only show in America because my research here right now has not been published. So for another region, I think I will show it later after my project is finished and published. So this is my research that I've done, and I use Nextflow for dealing with my analysis in my research, because my analysis or research is dealing with so many data and very, very big data, and I need to perform it in automatically and in parallel. So I use Nextflow to build my pipelines, and they can perform the analysis I think less than 2 weeks to perform the assembly for a 1,000 genome or transcriptome data. So yeah, that's all is my presentation. I give my back to Yola.
Yeah, thank you, Mr. Fahmi, for the presentation. It's really interesting because in here we have 2 essential information for everyone. So for the first, we know that we have a new open source web server called Nextflow. So it has the function as a manager, right?
Yes,
Yeah.
yes.
So we can manage our methodology or flow to creating the methodology for our purpose. And the second, from the research of Mr. Fahmi, here we know that the loss of biodiversity, it essentially can have a big effect to the, how to say, from the data and then from the spatial of the plant. Yeah. So in here, before we are going to the next session for Q&A, so please for everyone that you have any questions, so you can raise your hand by using the menu in Zoom, or maybe you can put the question into the chat. So in here we have one attendee that already raised the hand. So for Mr. Maulana, please go ahead.
Okay. Hello. Am I
Hello.
already in the screen now?
Yeah,
Yes.
it's already clear. Please.
Okay. Okay. Thank you. Wow, this is a very interesting things, right? Because okay, this is about the next flow. I never used it before, because normally I work when I set up my pipelines. Normally I do it by myself, right? But nowadays we have some like it's already deployable ones. So Mr. Fahmi, I want to ask about these pipelines that you present about the next flow, because we never used this before. And is this pipeline that you offer that you present is already deployable in form of the Docker image, or you it's already contain the environment, contain some customized applications that already pre-carried inside in the Docker image, or how you explain the next flow? Because we not yet have use it yet, but maybe you have some experience on it. So can you tell us about this more, maybe?
Okay, thank you. Okay, so in the case of Docker images, so in Nextflow you can like specify which Docker image, or for example, if you want to perform analysis using a specific program and you don't want to like install it on your local PC, you can just like define the image of that tools in Docker image in the Nextflow. So you can just define the Docker image in Nextflow, and then Nextflow will like retrieve the images from Docker, and then it can run the program in your in the Nextflow. So yeah, Nextflow can be combined with Docker and another containerization. For example, if you are familiar with Singularity or Kubernetes right now, you can also combine another containerization with the Nextflow very easily. And yeah, you don't need to install all the requirements. You just need to define what Docker images that you want to use in the Nextflow. Yes.
So we must have the pre-existing Docker to run the Docker image.
Yes.
Or
Yes.
okay.
You need the pre-existing, and I think there's so many Docker images that is available in the Docker websites. You just need to write the names of the Docker images that is available in the Docker's repository, and then you can just define it in your Nextflow, and then they will download the Docker images from the repository automatically. So you don't need to like download it one by one. You can just need to define it, and they will be run it automatically. It also can be combined with the Conda. So Conda, if you are familiar with Conda, you can like if you have so many tools that you want to use, you can define it in the Conda, and Nextflow can be combined with Conda, and then Conda will download all the tools automatically for your programs.
Okay. I think for this discussion will be like very long about when we talk about the Nextflow. Maybe can you give us like some contact so we can contact you more, so we can learn more.
Oh
on
yes,
this.
yes. Of course, yes. You can contact me in this email. So I'm available from in this email. So fahmi@gq.ac.gp. So
Okay.
yeah, if you want to discuss more with me, yes, you can contact me. Yeah, you can also contact me by WhatsApp. So if you want to chat more, so you just need to contact me in this email, and then yeah, I will give you my number. So and we can discuss it later.
Okay. Thank you very much then.
Yeah.
Thank
You're
you.
welcome.
Thank you, Mr. Maulana, for the question. So maybe anyone, if you still have any question, you can drop the question into the chat box. Or maybe while we are waiting for the question, I just want to continue the question from Mr. Maulana. It's about this open source, because I realized that this is the AI-based open source, right?
Yeah, you can combine it, the AI in it. It's just like the workflow managers that you can combine all tools within this workflow manager.
Yeah, that's the thing that I want to know. Because before we are going to combine this Nextflow with the Conda or Docker or another platform, so could you please tell us about the basic principle or concept about this Nextflow? Because maybe it can give us the insight more, so we can combine it with another platform just for our purpose for research.
Yeah, the basic things is about writing a pipelines. So here, yeah, in this in the modern bioinformatics, so you often deals with so many tools that, for example, if you want to perform a phylogenetic analysis, so first I think you need to have the sequence, and then after that you need to align the sequence, and after the aligning you need to like perform the phylogenetic analysis, whether in the maximum likelihood or other methods. So there are some kind of steps in the bioinformatics, right? And they often use different tools to perform. For example, in the phylogenetic, there is one tool called Mega, but it's not so comprehensive. So if you want to perform a comprehensive analysis for the phylogenetic, you need like a MAF for the alignment, and then for the phylogenetic analysis, you can use RAXML. So there are two spirits tools here, and if it's only two, you can run it manually using like a GUI. You just click, insert your data, and then you run, and then you get the output, and then you insert to another tools. But if the pipeline is very complex, for example here, for the assembling the genome data, so you often need to perform the analysis in many tools that is different. So they are separated. So for example here, for the quality control, you need the FastQC for the quality control. And then there's also another tool called Trimometrics to trim the sequence that has bad quality. And then after that, you have another downstream analysis that you usually perform in another tools. So it will take a lot of time to perform it manually. So you often need to write the script. So you just can run it once, and then they will automatically flowing your data into one tools to others automatically. But if you have so many data and you want to perform some kind of a parallelization, for example, if you have like a 100 data, so if you run it one by one, for example, one data, after finish, you run another one data and finish, it will take a lot of time. So you often need to perform like 10 data in parallels. So for example, 10 data, you run it 10 data in parallels. After finish, they will also input another data in parallels, and etc. It will boost your time in performing the analysis. In my case, I have like a 1,000 genome and transcriptome data. So it will take a lot of time because the size of data is around like 100 terabytes. And to do the analysis, I need the parallelization. And Nextflow can perform the parallelization automatically. If you write the pipelines or you have the Nextflow pipelines, they will run it automatically in parallels if you have so many data. And because of that, you can boost your time in performing the analysis. So that is the basic concepts of Nextflow. So you write the automatic pipelines for these pipelines. You write the pipelines for these pipelines, and then you can run it in parallel, so it can boost your time.
Yeah, thank you. So that's really clear, because in here we know that by using this platform, we can do the research in the parallel, and we can minimize the time for the analysis, because previously when we want to, like, for example, when we have the sample for the SARS-CoV-2 the previous time, if you want to analyze the sample for the SARS-CoV-2, it takes time, because we have 1,000 samples for the genomic. So I think this Nextflow, this platform is really helpful for us, especially for the phylogenetic.
Yeah.
Yeah.
Okay. Yeah. For example, another example, if you want to perform a docking. So right now, I think so many researchers in Indonesia performing a docking analysis. If you want to perform a docking analysis, for example, like a 1,000 or like a 10,000 proteins, automatically you can perform it using Nextflow. So it can run like a 1,000 or like more data in parallel. So yeah, you will get the reports, and then you can have the result, and then you can compare the result from that thousands of data. So yeah, I think that is the concept of Nextflow, like performing a very big kind of analysis in parallels easily. So, and there are so many scripts. I think if you not familiar with writing the script, there are so many pipelines that is available. You just need to download it in the GitHub, and then you can just follow the instruction, and you can run your kind of analysis seamlessly.
Okay, thank you, Mr. Fahmi, for the answer. And now we are going to the question from the students. And here we have 2 questions. So the first question is from Christopher. I think he really curious about the Nextflow, because he asks about how we can study this Nextflow in efficient way, and maybe how long it will take for us to analyze the sample.
Yeah. For my experience, like there are so many workshop and pipelines or documentation that is available in the YouTube. You can just use YouTube to conduct to study. And then I think there also has a Gitpod platform in which you can register in free, and then in that platform you will have some kind of steps to learn, and you will be given some scripts that you want to learn, and then they will give you an example of the case, and then you can there are so many platform that you use, and mostly in free, so you just like need to Google it, like Nextflow documentation or Nextflow class, and etc., and you will find so many platform to learn. And for my case, like I learned it in I think 2 months. Within 2 months, I can write the pipelines, because the concept, the basic concept is very easy. Yeah, it's just based on this input and output. So if you have bioinformatics command line, so you just need to write this input and output, and you can write the pipelines. So for example, from this output, if you want to use this output in another process called index sample here, you just need to write like the input from this output, and etc.
Yeah, thank you. Thank you, Mr. Fahmi. So maybe in the next time, or maybe next semester, I will try to enrich the bioinformatics class with this Nextflow, perhaps. So maybe we just need to prepare it first. Thank you. Okay, so now we are moving to the second question. It's from Zikri. It's about your research, Mr. Fahmi. And in here, he asks about the lost biodiversity that can be a threat for us. So what actually that can we do about that, so we can like minimize the threat of the lost biodiversity, and should we use less herbal product to protect the biodiversity or something?
Yes, there are, I think like there are two major steps. First is identifying the region that has the biodiversity loss. For example, here this research identify which region that has that the region has many threats. So first we identify the threats, and other things that we want to analyze is whether in that region there are so many potential. So in that case, that's what I do. So I want to identify the potential of the regions. So in this case, I want to identify the potential of medicinally associated enzyme diversity in a region. So for example here, so the heat maps with in color yellow, they have some kind of potential for the diversity of its enzyme that is associated with the natural products. So after identifying the threats and identifying the potential, we can like make the policy whether like this region, if they have so many threats, we need to perform the policy to counter the issue. So to protect this biodiversity, we need to kind of like make this region is like as a conservation area and etc. And that's what we can do to minimize the loss of biodiversity. We should identify first the threats and the prospects of our regions.
Yeah, thank you, Dr. Fahmi. So it's been in the end of this research, it's not only about the publication, it's also about how we can build the policy for the government to protect or to conserve the biodiversity of the planet. That's a big thing. Yeah. Thank you, Dr. Fahmi. I think there is no question anymore, so maybe we will move to another session. This is another session for the speaker 3 and speaker 4. And before we are going to Mr. Johan as a moderator, so I would like to acknowledge Mr. Rezi and Mr. Fahmi for attending this session. We get so much knowledge and update information about the benefits. So thank you, everyone.
Yeah.
And now I will move to Mr. Johan, please.
Yeah. Thank you, Ms. Yolanda, and also our notable speaker, Mr. Resi and Mr. Fahmi. And now let me continue the second session of this global webinar. First, I want to greet our third speaker, Dr. Murthy. Are you there?
I'm here. Can you hear me?
Yeah, greeting Dr. Murthy.
It's been a long time.
Yeah, it's been a long time also. Then let me introduce first our next speaker background. Dr. Satya Murthy is a deputy principal researcher at Insilkuchen in South Korea, and he has over 13 years of research experience in various roles, including as a postdoctoral fellow at the Korea Institute for Advanced Study and Kyunghee University in South Korea. And I'm also want to mention that he is also my mentor back then in the Kyunghee University, and also as a senior research fellow at Tamil Nadu Agricultural University in India. His research interests include genomics, next generation sequence data analysis, the novel transcriptome, and the genome assembly, comparative genomics, metagenomics, and integration of multi-omics data using the machine learning. And Dr. Murthy has received several awards and grants, including the Kyunghee University Presidential Scholarship, Best Researcher by the Korean Jensen Center for Most Valuable Products, and Best Oral Presentation Award from Korean Protein Society. So yeah, until now he already published over 55 articles and one book chapter with total more than 1,000 citations. So let me introduce Dr. Murthy, and yeah, this is your time for your presentation. Thank you.
Okay. How do I share my screen? Okay. Can I start my presentation now?
Sure,
Um,
the time is yours.
Can you hear me?
Yeah, we
Yeah.
clearly hear you. Thank you.
Yeah, it's
Okay. Okay. Thank you for your nice introduction. And it's been a long time to see you as well. So since I've been invited to give a presentation on AI and biotechnology, I choose my topic on benefits of decoding medicinal plant genomes. Since I'm long been working on medicinal plants, so I took this topic to explain more clearly to the students. So why I choose this one? Once again, yeah. Since you are given a topic on artificial intelligence in biotechnology, so as for my experience, I can say the artificial intelligence is nothing but it's a tool for a biotechnologist to bring up the knowledge from the massive data which present in public data sets. So here our role is just to convert the unstructured data to the structural model using the biological components here. One second, I will change pointer option. Okay, so I will use my mouse pointer as the pointer. So here we are using a biological components to convert the structural data, unstructured data to the structural data to create a model with the help of features. So these features comes from biological components. And from this model, we are going to get the logical inference. That means based on our model, we are getting a reasonable data that going to give us a knowledge. And this knowledge taken forward to the wet lab experiment. So this is the flow when you work on artificial intelligence. That means you are taking a help of AI tools. The rational method, directly we are converting, we are taking the unstructured data with our heuristic knowledge, we are taken forward to the wet lab experiment, and we are circulating here. This will take a lot of time and cost, and the success rate is low because we are just going into one components. That means we might have some assumption on something, and we are taking that directly to the wet lab experiment. And mostly the 50-50 chance, 50% we will get a positive result and 50% we will get a negative result. But what we can do now, we have plenty of data in a public data set, so we can create our model based on our requirement, and based on the result, based on this logical inference and the knowledge, we can go forward to the wet lab experiment. In this workflow, we will get more success based on our targeted problem, and we will reduce the time and cost. So this is the first beneficial thing we will get from artificial intelligence in the biotechnology field. So next, the main problem is while converting the unstructured data to the structural data is the missing data. Mostly we have a problem, but we don't have a data in public data sets, since the NCBI or GenBank have massive data. But when you go into the specific problem, the straightforward, you will face the missing data. So here, the problem I taken is traditional medicines to drug discovery. Why I took the traditional medicine to the drug discovery? Because in ancient time, and our Asian people are more apt or mostly use this medicines to treat some diseases. In case of Indonesia, it's called jamu, and in case of South Korea, it's called Oriental medicine. but the root which caused these medicines are from Chinese traditional medicine. But when I look into the Chinese traditional medicine theory and the Oriental medicine theory, the Oriental medicine rooted from Chinese medicine only because of this ginseng, because the ginseng is equally traded to gold. So at that time, most of ginseng cultivated from the Korean peninsula and taken to the Chinese market, and it's sold for equal to gold, and it's traded equal to gold. At that time, this is a big, this is the one of the valuable commodity to the traditional medicine people. Even when you see this Chinese medicine literature, they have 2050 plant-based formulation. Among those, 80% of formulations includes ginseng. So this is the history. From this history, how we can take it forward to the modern medicine is very important thing. So I taken this problem and I'm trying to find some solution. So the thing is, mostly the literature, the traditional medicine literature written in their own scripts, like a Chinese or Korean or Egypt or whatever in Indian languages and in Indonesian language. It have their own language, but the interpretation of that formulation for the global community is quite hard. So as now we are depending on the modern medicine, we need to move forward, take those literature and those knowledge to the modern medicine with evidence-based medications. Okay. Now, even cancer therapeutics depends on combinatorial drugs. Do you know what is mean of combinatorial drugs? Is a combination of multiple drugs. Now, but 1:1 will not work. So now even modern medicine going into the combinatorial drugs. But I was surprised the similar principle was used in our traditional medicine, but they don't. But they might know what are the components present in those extracts, but still we don't know what are the components present in those extracts. So this is a direct connectivity between combinatorial drug and the extract-based traditional drugs. So I want to find the relationship among this, among these two components. Because why I am working on this problem in the sense, most of the chronic disease people and the old age people are highly dependent on the traditional medicine or the supplements which are available in the market. So they should know what the supplement they should get for their illness. I'm not saying that this traditional medicine directly cure the chronic disease or the mortality-based disease, but I'm sure that it could, this research could help the people who depends on the traditional medicine or based on the supplements. So when I look into history, it started, the drug discovery started from the 18th century or 19th century, is a 19th century. First, they started with morphine drug. First, they discovered the morphine drug from the narcotic plants called Papaver somnifera. From then, in the whole 19th century, where started to isolate the morphine drug is the first period. And later, the second generation, they started to discover the vaccine and the receptor-based drugs. And third generation depends on hormone, vitamins, and those stuffs. From then, now at present, we are involving in gene editing technologies to cure the disease directly inside our body, rather giving any oral drugs or any external supplements. We are now ready to cure our disease within our body by editing the gene. But it's one, the caste wise is not affordable for the developing and underdeveloped countries, since the oral medication is the best choice for them to treat the chronic diseases. So this research is more important, and we need to move faster. As in the previous presentation, Mohammad Famik said that the huge diversity loss, the plant diversity loss could also save from this research, this kind of researches. So when you see the scenario around the world, most of people working on medicinal plant to identify the novel drugs for treating the human diseases. Among those, the country like China, India, and South Korea, and USA are working more. As for Mohammad Fahmy information, yes, US people are exporting more plants from around the world, and they are utilizing the resource, but they are the another problem is it's destroying the plant diversity. To save this, we need to go more in detail to find out the exact phytochemical and the enzymes present in plants. We need to identify those very clearly. That's my objective as well. So when now I'm going into the basic of bioinformatics. Why we need to do these things? Because our body is organized into the once again, our body is organized into the organs, and the organs are made up of cells, and the cells made up inside the cell we are going into the chromosome and DNA and nucleotides, and these nucleotides were formed into the atoms. So here, when you go to the doctor, doctor can you can explain about your phenotype. I have cold, I have this, I have stomachache, or this is called a phenotype, and they used to examine the phenotypes and the signal which you got. When you go to this next level, after doctor observed your symptoms and he referred some medical imaging system like CT, MRI, and those scanning system, we are getting another set of data. After then, we are going for some biochemical test, that means the urine or the biopsies, liquid biopsy test, like a blood test, urine test, and you can go with another cultural in vitro cultural tests to identify the pathogens. And beyond that, we have to go into genome level or in genetic diagnosis when you are getting some rare symptoms. So this is called genotype. So after that, once you understand which enzyme or which protein causes the disease, or that could be a causative to the particular particular disease, then we need to take the protein. So this is the overall system, the system organization, and how the system's taken into data, and how this data can be imported or subjected to the machines to get a knowledge. Here, I am highly dependent on connecting the genotype and the phenotype. This is called knowledge mining. That means once you're able to connect the genotype and phenotype, sure, you can identify the key enzyme or key disease marker, and then based on the marker, you can understand how it could treatable. So whether you have a known known system to treat it, or you need to find a novel medicine to treat the disease. So this is a simple phenomena working on in the medical industry. So the another component, if you see, after father of genetics, the Mendel in 1865, he started to devise the rules for genetics. Then Sanger in 1977, he invented how to sequence the nucleotide and protein. From then, in 1990s, US government started the genome sequencing, the human genome sequencing, and in 2003, they released the first human genome sequencing, the whole genome sequence to the public. So it takes almost a century to come up with the whole genome sequence. After then, within 10 years of time, or within 15 years of time, we get plenty of human genome resources around the world. Even the individual countries started their own genome project to create more than lakhs and lakhs of genomes to treat the human more precisely. So this is how around the world, the human community started to decode their own genome to find out more treatments for their illness. So similarly, when you look into the gen bank, the public data sets also growing enormously. So from this, we need to find more knowledge. That is our goal in future. In future, the only reason all this happened because of the genome cast and genome sequencing technology. Okay, now even if you have $100, you can decode your genome, your own genome, and you can compare those with the public data sets. Then you can understand what is your genome and how this, you can prevent or you can treat yourself. That is our future future objective. So these technologies all came from the human studies, but this technologies taken forward to the plant system. So far, there is around 700 genome sequenced all around the world. But these genomes are highly based on the vegetables, fruits, and other set of crops, but mostly the medicinal plants genomes are still missing. So here I'm taking a help of bioinformatics, and when you see into the artificial intelligence, these components play parallelly. Okay, in case of bioinformatics, is just these are the components: biology, information technology, mathematics, statistics, and computers are combined together to create a knowledge from the data. The similar thing happening in artificial intelligence. Once you have a defined data, that means the training data sets, you can get a knowledge with all these components. Again, that mathematics, statistics, and computer science is the different discipline of AI. So the element of bioinformatics with the respect of genomics is we are finding the domains, motifs, patterns, mutations, variations, and signatures. These are called features to the machine learning. So here, once if we know what is genome and what are the components of genome, those are the features for machine learning. If you're able to create a new features to your machine learning model, you will get more precise or more clear knowledge from the genome. So this is the genome-based components which used in bioinformatics. And from these components, we are just the whole bioinformatics process is finding homologous, orthologous, and paralogous to connect those genome to the other genomes into the networks. That means linear form to complex network. This is the whole bioinformatics process happening. And when it comes to metabolomics, still, even Johan have this experience, I guess. So when you use the whole sample for metabolomics data sets with the help of MS, most of the components are unknown. So here the problem starts. We cannot annotate the complete metabolome data, what the instrument given to us. But in case of genome, if you have a genome, you can annotate it. You can get some knowledge from the genome, and you can connect the genome to other genomes. But overall, you can understand 90% of genome components with some knowledge. So I'm trying to connect these two components, genome and metabolome. In this case, when you go into the model organism, you will get almost 90% of knowledge from genome and around 50% of knowledge from metabolome, so that you can connect and you can get the reactions, and you can clearly able to figure out which reaction is going on to create this metabolite or synthesis particular metabolite. But when it comes to the noun model, that here I can say example is medicinal plants. We cannot say that because most of the medicinal plant doesn't have a genome, and most of the medicinal plant doesn't have metabolome data. So here we need to take most of the biotechnologists, one who are interested in creating a novel drugs, have to focus on this area, because for how to say, for example, in case of edible crops like rice or tomato or sweet potato, they have a consortium. But in case of medicinal plant, we need to take forward, and we need to make a new consortiums to speed up this process. So that's why I'm taking this presentation to the new coming people into biotechnology. So this is an example, just an example, because of Corona is a pandemic. Immediately there's our scientific community have to respond to the disease, and we need to find out some drug or some vaccine. So they relaxed the rules, and now we created a vaccine in 10 months of time. Although the vaccines have side effects and a lot of things, but still now after within 10 months we got a vaccine, and within 3 years we have vaccinated, and now we came back to the normal state. This is the help of the technologies what we developed, that called genome technologies, because as soon as the pandemic arrives, we got a genome for coronavirus, and we got a structure for spike protein. After this spike protein, the spike protein was screened for various drug with the help of a drug repurposing method, and they find out some sterile drugs, and they started treating to the public. So similar thing, how it happened, similar thing can happen in a medicinal plant research, because the another step is in protein structural prediction, AI dominated, because now, because of the AlphaFold, we have the whole proteome, a structural proteome in our hand. So which means you can screen the individual drug to the whole protein data set, to the whole human proteome, like next flow pipelines. Because as he mentioned, you can screen a single drug to thousands of protein in parallel in the same time. So here my claim also the same. So now if you able to find out a novel drug from a medicinal plant, you can screen that metabolite to the human proteome, the whole human proteome, instead of single protein or single receptor. So this is the advantage of using AI in biotechnology, because here the AlphaFold protein structural database is free to public. And if you have a component and you have n number of tools like similar to Nextflow to screen those metabolite to the human proteome, but still the missing thing is the genome component for the medicinal plants. So here I take 3 examples. One is ginseng, another one is platycodon, and another one is papaver. These are my researchers. Last 3 years I working on this problem. So here, as I said before, in 19th century, first panacoline is the basic component. That means the Panax, in the Panax ginseng, we have the compound called ginsenoside. This was invented in 1854. Later, we have numerous ginsenoside belongs to triterpenoids. So the major biosynthesis pathway is the downstream pathway of isoprenate pathway. So in this pathway, we have to, our task is to find out the novel cytochrome enzymes and carbohydrate enzymes. That means glycosyl transferase and glucose transferase enzymes, which can fit into or which can implement to enhance this biosynthesis pathway. So around 2010, we use only EST to find out the cytochrome enzyme, but that time we have only we able to find only the clan 71, 86 and 72. But now, after we got a genome sequence, we able to find out so many cytochrome enzymes present in ginseng genome. That is the benefit of having a limited transcriptome and the whole genome. So here, when it's come to ginseng, not only the Panax family is called as a ginseng, the other ginsengs like pseudo ginsengs called Genostoma pantapylum is a poor-man ginseng, Codonopsis, platycodons are the poor-man ginseng, and Indian ginseng is Withania somnifera. These all have the triterpenoid components which could treat multiple diseases like anti-cancer, antimicrobial. It been studied for so many diseases. So I want to connect these plants, and I want to find out the novel cytochromes or common cytochromes and glycosyl transferase enzyme from the genome. So this is an ongoing study. Now we have plenty of Panax ginseng genomes, but still we have some missing genomes like Indian ginseng and other got a ginseng genome by last year, and still we are working on Indian ginseng genome to find out this cytochrome enzyme and glycosyl transferase enzyme. The another example, as I shown before, the first drug discovery started from morphine drug that came from this narcotic plant, and this narcotic plant can be used as ornamental plants and the narcotic drug plants. So to differentiate this, we created a transcriptome data and metabolome data, and we connected the cytochrome database, and we found the novel cytochrome enzymes to enhance the isoquinone alkaloid biosynthesis pathway. If you see clearly, some enzymes were only present in the pepper plant and not present in other plant. So we taken this cytochromes to the experiment data set. And after then we created a metabolome, a targeted metabolome, and we compared the expression, transcriptome expression and the metabolome enrichment. Then we are combining these two, and we are creating a data set to implement the machine learning model. Then next, the platycon flowers. Based on the flower colors, there is a chance of some enriched metabolite, some different metabolites. So we the initial step, we collected large diverse planticodon plants genotypes, and we predicted the plant colors from the genotype. Now we are looking forward to predict the metabolites from the genotype. So it's our next step. So those data I cannot show here because still we are working on it. So I'm just limiting the data here, and this way we are converting the genome components and the metabolite components, and we are importing those components to the machine learning models, and we are creating an effective model to predict various features like which metabolites are enriched in which plant, and within a plant how these metabolites were synthesized in which stage of the plant. So this kind of things we are working on. And as a role of bioinformatician, from the data, we need to create a theory. So that's what currently I'm working on. From genome and metabolome, I'm creating a database to get more precise knowledge about the medicinal plant and its supplements. So this was my story, and I am asking the newcomers to develop more genome for medicinal plant and more metabolome data in the same time scale. And if you're able to connect this both, sure, you will come up with the new drugs and you can help the human community. Thank you. Thank you. If you have any question.
Yeah, thank you, Dr. Buti. Yeah, nice to see your closing picture here. I'm also there.
You are here.
Very long time ago. Nice to see you again, and yeah, you update us with many insightful information nowadays. How we can use many tools to easiness, yeah, of our research. And I also like that the conspiracy theory of unicorn. Thank you. Okay, folks, we hope you already enjoyed this webinar so far, and I also encourage all of you to ask the question or drop the comments in the chat box, and we will make sure to utilize to answer as many as possible during later the question and answer session. Thank you, Dr. Muti, for this session. Please hold up for the next speaker. I would like to greet Dr. Oscar. Yeah, Dr. Oscar, are you there already?
Yes, I'm here. Can you hear me?
Yeah, we heard you.
Yeah, it's already clear.
Thank you.
We pin your so. Oh, please, Miss Yolanda, can you help me to pin the next speaker?
Yeah, sure. Please wait.
Yeah, Dr. Oscar. Nice to have you here also. In the last but not least, in our Ubaya Global Webinar session, please let me introduce some of your research background in a few minutes. Wait.
Okay, thank you.
Okay, here, yeah, Dr. Oscar already working. Yeah, nowadays, yeah, the current affiliation with the University of Tsukuba, Creative School of
Yes.
Life and Environmental Science in Japan, and where he holds also a position as the postdoctoral research fellow. And the source that I found, Dr. Oscar has more than 10 years of research experience in various fields, including molecular biology, biochemistry, and biotechnology, with a particular focus on the plant biotechnological and plant microbe interaction. Wow, this is interesting. Has overlapped with my research focus also. Nice to meet you. And according to the source that I found also, it's already published more than 25 articles, which has been cited more than 600 times, and giving him an HN index of 13. His research interests include the plant microbe interaction, plant biotechnology, functional genomics, and metabolomics. He also worked with various research projects, including the transcriptome analysis, gene expression profiling, metabolic pathway analysis, and plant transformation. Now, we'll hear the presentation from you, Dr. Oscar. Time is yours.
Can you see my slides now?
Yeah, it's already showed.
Okay.
We can see. But
Okay, so
okay,
first,
okay, thank you.
First, I would like to thank the organizers for this opportunity to present in this webinar. As you have heard, my name is Oscar Mitalo. Many people just call me Oscar. And today I will discuss about something different from other presenters. And it will be about the post-harvest physiology of horticultural crops. And in this talk, I'll focus more on fruit ripening, the role of ethylene and low temperature. So to begin with, let me start with a small introduction, since I'm new to many people here. I'm from Kenya originally, and this is where I did my Bachelor of Science in Food Science and Technology. And then after that, I worked for around 1 year, after which I moved to Okayama University here in Japan. And, in this university, I did my master's and PhD studies in post-harvest and physiology and technology of fruits and vegetables. Right now, I'm working at this the University of Tsukuba, and this is our building. It's a very small building. Most of this area is just the farm, and I'm working as a JSPS postdoctoral research fellow to date. And so just for a brief introduction, many horticultural crops include mostly fruits, vegetables, and flowers. And most of these crops, they are known for having being a good source of nutrients, having high water content, as high as some of them might have as high as 95%. And these crops also, they are easily damaged. In other words, they are very perishable. And this damage begins immediately the crops are harvested. And one important characteristic of these horticultural crops is that they are still alive. And by this I mean they can still breathe, they can produce heat, they can lose their moisture, and they can get sick. They can get infected by pathogens, and they can, after that, they can even die also. So there are three main important biological processes that affect the quality of fruits and vegetables, and these are listed here. We have respiration, that's the main one, and then the transpiration or water loss, and then lastly we have fruit ripening. So these three processes, especially respiration and transpiration, they are the most contributors to post-harvest loss, because they are using up plant resources. And when these fruit or vegetables are still in the parent plant, once the resources are lost, they are easily replenished by the parent plant. They can easily get back from the parent plant. But when you harvest the crops, they are on their own, and once they lose the water or they lose the starch due to respiration, it's never replaced, and this is the reason why the deterioration begins. And today I will focus mostly on fruit ripening, because this is what I've been researching since my master's. And fruit ripening is a very important process because it is what makes the fruit edible and palatable for our consumption. Because during this process, the fruit becomes sweeter and they lose the acidity. They become softer and they gain attractive color. And some fruit develop pleasant aroma. But the major shortcoming of the ripening process is that it can, if it's uncontrolled, it can easily reduce the post-harvest life. Because when the fruit ripens, the next step is senescence. It fastens the senescence process. And then fruit ripening also increases the susceptibility of the crops to disease, because once they are soft and they become sweet, these conditions are very ideal for growth of pathogens. And also, ripe fruit are very difficult to handle, especially the soft ones. When you handle them, they can easily spoil. So it's very important to know how this process is regulated, so as we can control it, so that the fruit can ripen when we want. And many years of studies have shown that this plant hormone ethylene is the main regulator of fruit ripening. And ethylene is a very simple gaseous hormone, and it's a hydrocarbon with the 2 carbons and 4 hydrogen atoms. And it's synthesized naturally in the plants via this pathway from the amino acid methionine. And the key enzymes include SEC synthase and SEC oxidase. And the plants have several receptors that can perceive ethylene signal, and then these receptors transmit the signal several components, which can eliminate in the up or down regulation of ethylene responsive genes, including those genes that regulate fruit ripening. And for fruit, we have 2 systems of in whole plants, we have 2 systems of ethylene production. And the first one is called system 1, which is responsible for the basal levels of ethylene production. These ethylene levels are responsible for mostly for defense and others, some other biological processes. and it's produced mostly during growth, the growth stage, and in the vegetables, in the leaves. But we have the system 2, production. This is kind of autocatalytic. It's a massive ethylene production. And this ethylene is only produced during ripening in some fruit. And it is this ethylene that regulates the ripening process. Based on this classification, fruit are classified into 2 main groups. We have the climacteric fruit, and for the climacteric fruit, they derive their name from this peak here. It's the ethylene increases rapidly until it reaches a climax, and showing this pattern during ripening. And this in this fruit, the ripening process is primarily regulated by the hormone ethylene. And the second major group is the non-climacteric fruit, including the citrus, strawberries, and grapes. This fruit, they continue producing system 1 ethylene even during ripening. So in other words, they do not require ethylene to ripen, although ethylene can regulate some ripening processes. But for this fruit to ripen, you need to leave them on the tree, because if you harvest them unripe, they'll never reach eating quality. And in early 1990s, this chemical called 1-MCP, 1-methyl cyclopropene, was developed. It's a synthetic chemical that has a same binding affinity as ethylene. It can also bind to the ethylene receptors. This chemical, unlike the ethylene binding, this chemical binds, it has a high affinity, and its binding blocks ethylene from binding to the receptors. And in this way, it blocks the ethylene signaling pathway. And this 1-MCP is gaining use of commercial use to extend the post-harvest life of many fruit. And as shown here, for tomatoes, if you treat them with the 1-MCP at the turning stage, it inhibits ethylene production, and the ripening process also is inhibited. Also, cold storage is widely used in the post-harvest technology of horticultural produce because of its broad spectral inhibitory effects. Cold storage, low temperature can inhibit fruit ripening. It also inhibits respiration, can also inhibit the senescence. And in addition, it also inhibits the microbial growth. It has very many advantages. That's why many people use cold storage during post-harvest handling. But the problem with the cold storage is that it can lead to some chilling injuries. So in some crops, especially the tropical and subtropical species. Then, I don't know if have you ever heard of kiwi fruits? Kiwi fruits are very popular. It's a popular fruit, national fruit for New Zealand. But it originated from China. This fruit shows a very strange ripening phenotype because generally the hormone ethylene can trigger the softening, the ripening process. As shown here, fruit treated with the propylene. Propylene is an ethylene analog. That's the same function as ethylene. When you treat them with the propylene, it induces the rapid softening. The softening is inhibited by 1-MCP, strongly inhibited by 1-MCP treatment. And this is a typical climacteric ripening pattern. But the problem is during on-tree maturation, this is the phenotype that is shown by kiwi fruits on the vines. And this research was presented by researchers in New Zealand in 2011. They show the growth patterns of the kiwi fruits. The green line, I want you to focus on the green line because it's showing the changes in firmness. And mostly at around this point here, that's where the softening occurs. And the brown line here shows the ethylene production rates in the fruit. So the fruit starts to soften. Most of the softening is occurring when ethylene is not detectable. And ethylene starts to be produced when the fruit have already softened. So the question has been like, what really causes the softening, this softening here? And that's what we try to address in our lab. So another problem is during cold storage. As I mentioned earlier, cold storage is widely used to extend the storage post-harvest life. But during cold storage at 0 degrees Celsius, at different ethylene concentrations, you can see we have 1 ppm. Red indicates 1 ppm, blue indicates 0.1 ppm, and the purple 0.01 ppm. And in air, it's the green line. Air means no ethylene, but the researchers assume that it's 0.001 ppm. That's not detectable. And it's shown here. When you have higher concentration of ethylene, the softening occurs faster than when we have no ethylene. And this rapid softening is obviously because of the ethylene hormone. But when you look at from another angle, the fruit in air, stored in air, they have softened considerably after 12 to 14 weeks compared to at harvest. So the cause of this softening, even in air during cold storage, is also not well understood. And there's a lot of debate regarding that. And so to address this issue, first we try to avoid eliminate any ethylene sources during storage. And you know, for kiwi fruit, they are affected by many fungi. Particularly, the most notorious one is Pseudomonas. And when the fruit are infected, they have these symptoms. And in addition, they produce ethylene. And the ethylene, when they produce, if one fruit produces ethylene, the ethylene can affect the neighboring fruits. So we tested this hypothesis using two different storage conditions. We stored in a group or separately. And after this, we monitored the ethylene production patterns of these two different storage techniques. And in the group storage, so here the different colors shows a different batch. I have 4 different batches. So in the same batch, you can see like they are showing, all fruit are showing almost similar. The peak, the ethylene peak is the same, almost at the same time point. And but when you look at it closely, you can see that in one batch, in this first batch, one fruit produces ethylene earlier than the others. And when we check this fruit, it is infected by disease. So only one fruit can affect the whole batch. But when we separate the fruit, we obtain this pattern. Most of the fruits that were infected by diseases were produced ethylene and softened earlier during storage, but many of more than 50% of the fruit, of the healthy intact fruit, did not produce ethylene and failed to soften, even when you store them for 75 days at room temperature. It was very surprising for us. And then based on that, we set up an experiment to compare the ripening pattern of kiwi fruit at during storage at 5 degrees and 20 degrees Celsius. And also we had separate groups treated with 1-MCP to make the fruit insensitive to ethylene. And what we observed was also very interesting, because the fruit stored at 5 degrees Celsius softened faster within 4 weeks, faster than those stored at 20 degrees Celsius. And furthermore, 1-MCP treatment failed to inhibit the softening induced by cold storage. So based on this finding, we hypothesized that low temperature can trigger ripening in kiwi fruit independently of ethylene, because 1-MCP, if ethylene is functional, 1-MCP can inhibit it. And we did RNA sequencing analysis to compare the effect of ethylene, that is propylene, and also storage at 20 and 5 degrees Celsius. And this is the heat map showing the genes, differential express genes that we found. And, shown here, most of the genes are regulated by the ethylene signal, but we also found genes in this area here. They are regulated by low temperature only. They are upregulated at 5 degrees only, but not at 20. They don't respond to ethylene also. So we call them low temperature specific genes. They are responding uniquely to low temperature signal. And we also just did a simple qPCR analysis to confirm the expression patterns obtained by RNA sequencing. And one pattern was shown here, that these genes are responding to either ethylene or low temperature. They can respond to either of the signals. But the interesting thing is that their upregulation by low temperature at 5 degrees is not inhibited by 1-MCP, meaning that ethylene is not functional. This upregulation at 5 degrees Celsius is independent of ethylene. And we also found another pattern, those genes that are responding to ethylene signaling only. They're not affected by low temperature. Then lastly, we also found, this is the genes I'm talking about. They don't respond to ethylene signal, but they are only upregulated at 5 degrees Celsius and not at 20 degrees. And 1-MCP also does not affect the. So based on these findings, we came up with a model where we showed that kiwi fruit can ripen in 2 ways. They have both ethylene dependent, and when ethylene is not there, the low temperature can induce the ripening also independently. So here in Japan, we have 4 seasons. It's spring in April, and summer in June to August, then autumn from around September to December, and winter is in December to around March. And the behavior of the trees during these seasons. During autumn, the plants start to change their color, the leaves color, and then they shed all the leaves. And the reason why they do this is just to survive. Because in winter, the conditions are very harsh. There's no rain, precipitation is less. So they need to remove all the leaves to lower transpiration. And so we observed the ripening behavior of kiwi fruit with compared with the changes in temperatures. So when the temperatures are high in September, the fruit are still not ripened. The firmness, they're still firm, but the firmness decreases gradually, and the sugar content also, the sugar content increases and the acidity decreases as the temperature decreases. So we hypothesize that this kiwi fruit, clever, they can sense that the temperatures, low temperature, environmental temperatures, so they need to initiate the ripening process, because ripening is not initially like for us to eat. It's a survival tactic. They need to ripen quickly to disperse their seeds. So maybe the onset of winter is a signal for them to initiate the ripening process. And this is also exhibited by the Satsuma mandarins. Here we have Satsuma mandarins and lemons. And you can see the color changes. In autumn, early autumn, they are still green, but the color changes gradually occur as the temperature decreases. The same phenomenon is exhibited by the lemons as well. So, citrus fruits are generally classified as non-climacteric because they don't require ethylene to ripen, but ethylene can trigger the color changes. Actually, ethylene is used in the industry to make uniform color for marketing. But the natural regulator during color change on the tree is not known. So we monitored the color changes during storage at 5, 10, 15, 20, and 25 degrees Celsius. And we observed that only fruit stored at 10 and 15 degrees Celsius initiate the color change, both for Satsuma mandarins and lemons. And 1-MCP, here, 1-MCP also does not inhibit the color changes, similar as we observed in kiwi fruits. So then we finally did RNA sequencing analysis also, and we also obtained 3 different patterns. The first pattern was this gene. You know, chlorophyllase is involved in ethylene-induced color changes, and it's only responding to ethylene, and 1-MCP can inhibit the changes as well. But no changes in expression are observed during storage. And interestingly, this gene does not show any changes in expression during on-tree, meaning that if ethylene is present during on-tree, then this gene also should increase, which will have an expression increase. But this confirmed that on-tree maturation is not regulated by ethylene. And the second pattern was shown by this gene, PPH. It's a phyphoetinase, has a similar function as chlorophyllase. And this gene responded to either ethylene and also low temperature. But the induction by low temperature is not inhibited by 1-MCP treatment. This gene is also shows a significant increase in expression during on-tree maturation, on-tree field greening, confirming also, showing more evidence that there's a role of low temperature in on-tree maturation. And then finally, this is what confirmed to us that low temperature, the fruit are responding to low temperature, because we have these genes that respond to, they don't respond to ethylene at all, but they only respond to low temperatures, but not at higher temperatures. And there's also expression increase during on-tree maturation. So to conclude, I just want to say that fruit ripening in the natural world is for survival, and plants have to find a way to attract the animals so that they can disperse their seeds. And you can check more information about in this article. Plants do this by changing their color to be more, to stand out, because if the color is still green, the animals cannot see them. The fruit have to stand out and easily identified by the dispersing agents. And those that cannot have color changes, you have to make a pleasant aroma to attract the animals, and also the softening or becoming sweet, just to survive. And so if you need more information about my research, sorry, I've been rushing, so I did not explain more detail, but you can get more in most of them papers are in this Google Scholar, my Google Scholar profile, or if most of them are open access, but if you cannot access them, you feel free to send me an email. I can send you the full manuscript. So with that, I end my presentation. Thank you for your attention.
Yeah, thank you very much, Dr. Oscar. So yeah, for providing us the fruitful information about the ripening. Because yeah, we thought until now that ethylene has the main actor of the ripening, but yeah, you and your group do the fascinating works to develop another kind of hypothesis about the ripening. Please hold on, because we will join together with Dr. Muti to this question and answer session, and also encourage for all of you, the viewers and also participants, in this Ubaya Global Webinar 2023 to ask the comments or drop your question in the chat box, or you can also raise your hand so we can see that you will have the question for our two speakers in this second session. Okay, Ms. Yolanda, please help me to pin the video for both speakers. Thank you. Okay, let me trigger first to Dr. Muti. Are you there, Dr. Muti? Yeah, for the Dr. Muti also, can we pin this one?
I think he already left.
He already left. Okay. So yeah, don't worry for the participant. Probably, yeah. I just remembered that Dr. Muti has maybe the urgent schedule for this afternoon, but we already have the his contact. So if you have any question regarding to his presentation, I would gladly to deliver your question. Okay, so I trigger first to Dr. Oscar about the free opening. Is
Yes.
utilizing or the usage of the MCP already common in the horticultural practices? Is already generally recognized as safe, or is still debatable for the practicable approach of this substance, Dr. Oscar?
Yeah, actually, one MCP is now being used commercially, mostly for avocados, and it's widespread in the US. But most countries have not allowed its use. Like here in Japan, we are not allowed to use it. But maybe in the near future, we'll be able to use it.
Yeah, exactly.
Yeah, the chemical actually it's not harmful because as you I did not show the concentration we use, but we are using only 1 or 2 ppm.
Yeah,
it's very small concentration, so it's not.
we know that. The unlisted is unlisted to the safe material is not same as the already toxic or harmful. It's not listed yet.
Not listed yet, yeah. But
yeah,
it's widely used in US. The US, yeah.
Yeah, we know that each country has their own regulation regarding the kind of chemical substance that use.
Yes.
And the important thing, you and your group in Japan already had the very strong evidence that there is another factor, another actor in this kind of the softening wrapping fruits, and it depends. It's concluded that there is low temperature specific genes. Until now, if it's not confidential, can you share more about how this kind of gene, the low temperature specific gene, already studied?
Sorry, can I get your question again?
Yeah, if it's not confidential for you and your group, can you share more about this? Maybe the low temperature specific gene already been studied, or is already annotated to what kind of genes that respond with more. You can either with the ethylene or also with the cold temperature.
Just a minute. I don't have them here, but the low temperature specific genes, they're very the wide functions. Most we have transcription factors there as well, and we have those genes that are involved in the downstream, involved directly in the fruit ripening regulation, like where is it? Can I share my?
Sure. You can share some slide if you want to.
Yeah.
Emphasize your explanation. Okay.
So, like here, look at these genes. This is SEO. SEO is involved in ethylene biosynthesis, And then we have pectin methyl esterase inhibitor, and we have pectate lyase that are involved in softening. And these genes this is involved in sugar changes, the starch degradation, and this is involved in gibberellin biosynthesis and metabolism, sorry. And these are transcription factors as well. And I'm now focusing on tomato. I've found genes also involved in epigenetic modifications like the DNA methylation, also responding to low temperature and alone, uniquely to low temperature. So these are candidates for further studies on the role of low temperature in post-harvest. Yeah.
I see. Yeah, it's quite interesting that you already make some list of the candidate genes that are responsible for it. And
yes.
I assume that this is only several notable genes. I assume that you already tried several genes to screen that one. Okay, I also want to invite again all the viewers and participants to share your thoughts or experience related to the topic being discussed this morning or this afternoon there. And your comments maybe inspire others and help to create more engaging and informative discussion. Please drop the comments or raise your hand to join this question and answer session. Okay, let me see. Yeah, we also wait for another question that come privately to me. Okay, and here, okay. In the practical approach, Dr. Oscar, can you give your thoughts how because this fact also fascinating because usually in practical area we deliver all the climatic fruits in the cold storage, but also cold storage become one reason the triggering the softening of the fruits. So how you give your maybe opinion or your approach to the best way to deliver or the logistic for the climatic fruits?
Yeah, actually, I understand the worries of the participant who asked the question because cold storage is the only way. We cannot avoid cold storage, but this is a dilemma. We have to make a compromise. Like, do you want the fruit to ripen with ethylene, or do we want them to ripen with the cold storage, with the low temperature? Because when you look at my data for ethylene ripening, the ripening in response to ethylene happens within 1 week, and the fruit have already softened. But for cold storage, they ripen after around 1 month, depending on the the cultivar. Some cultivars take longer, maybe 2 months. Yeah. So do you want them to ripen slowly and for 2 months or 1 month, or do you want them to ripen quickly within 1 week? We have to make these choices by ourselves. And also one more thing to consider is I published this data in Frontiers about the aroma production, because the kiwi fruit that ripen in response to low temperature, they don't have aroma, the characteristic kiwi fruit aroma. The esters are not produced, and that is a big problem. Yes, the fruit are sweet, soft, but the aroma is not not so good. And so we have to consider about these things when you are making a protocol, post-harvest protocol. I cannot just give you a definite answer. It depends on the you're using and the purpose you want to use it for.
Yeah, exactly.
Yeah.
Because in our probably tropical countries, the temperature, the high temperature, and the high humidity also become the key factor that make the fruit
Yeah.
become spoilers easily. So
Yes.
we have no other choice. We try to do the cold storage to prolong and to make sure the logistic becomes safe. Okay, I would like to read the other question that arrived to my personal chat. Dr.
Yes.
Oliver here. If you say that one MCPs are not yet recognized as the safe in several countries, is there any different like the synthetic materials or the differential from this substance that already used also with the analog or similar effect of this one MCP?
Other chemicals? Or
Yeah, probably chemicals or protein because the question also not pointed that one. Another substance that can has the similar effect. Is one NCP block the ethylene enzyme or what kind of the mechanism of one MCP?
If you look at my slide, sorry, I have to share again.
Yeah, it will be more clear for us also.
If you look at my introduction of 1-MCP.
That's slide 6, okay.
Oh, yeah, sorry.
Oh, another? Yeah.
This one here. 1-MCP acts by binding to the ethylene receptors.
Ethylene receptor, okay.
Yeah, it binds permanently because when ethylene binds to these receptors, actually it degrades them. And when it degrades them, it releases the CTR1 so that and the ion to go to the nucleus to activate the gene expression. But when 1-MCP binds, it binds permanently. And this binding does not trigger the signaling. So that's how it's able to block.
Yeah.
And there are many other chemicals that are similar to 1-MCP, but although they don't work exactly as 1-MCP, but they block ethylene signaling, but most of them are also, they're not allowed. So 1-MCP, yeah. So 1-MCP is the best so far.
Okay.
Yeah,
okay.
Because that's why it has been allowed in the US, and in Kenya, it has been approved for using avocado.
Oh, okay.
Yeah.
Because if we see this kind of the mechanism, we also can this is my personal opinion also.
Yes.
We can
This is,
the RNA interference to make the receptor expression become lower or nothing. So we just block in that receptor expression. What you can think about that?
The problem, yeah, you can do that also, but you have to worry about the effect on the whole plant because ethylene,
exactly, yeah.
Ethylene is not only for ripening, but they have ethylene for other processes as well, defense, and also for reproduction. The ethylene is needed, like for potatoes, ethylene treatment is used to induce germination.
Yeah.
Yeah.
Yeah. So the time point of how we treat also become matter because probably better to treat it after we pick up the fruits, not in the whole plant itself because it will disrupt everything, every other metabolism or mechanism that plant has. Exactly. Thank you for
so yeah, for me, I'm an ambassador of 1-MCP because I've tested it, and it's because we don't have, human beings don't have ethylene receptors.
Okay.
Ethylene receptors are unique to plants, so I don't think it can have any effect on human beings.
So that is specific for that blocking the ethylene receptor.
Yes.
Yeah, we already right at the end of this question and answer session. Thank you again, Dr. Oscar, for your insightful discussion for this session. And I would like, before we close this Ubaya Global webinar, we would like to have the photo session with all of you. Even though we are online, I would like to invite all of you to turn on your camera and give your best pose.
Just a minute.
So we can do the documentation for this. Please help me, Mr. Yolanda. Miss Yolanda to do the documentation.
Okay, thank you, Mr. Johan, Mr. Oscar, Mr. Bani, and all. Oh, Mr. Murthy is already here.
Murthy, you already came back. Later we will deliver all some of the personal question that already arrived in my chat box to your email. Probably you will answer at your convenient time. Thank you, Dr. Murthy. Okay, please join us, all the participants, so we can document this Ubaya Global webinar session.
Maybe I will take the picture by counting from 3, 2, 1. Cheese, everybody. Okay, that's for the first slide. So maybe I will move to another slide. Let's wait. Okay, so I'll run again. 3, 2, 1, and 2. Okay, thank you everyone for attending this webinar. I think this is the last session for our webinar. So I would like to say thank you for everyone that come here. Thank you, Mr. Oscar, Mr. Fahmi, Mr. Murthy, and all of the attendees here. So I hope that we can have the insight or inspiration from our speakers. Thank you, everyone. Thank you, Mr. Johan. We see you in
everyone. See you again in the future event. Have a great day.
Thank you.